In our final video in this series on glycolysis, we're going to investigate how the last phosphate that was originally added to our glucose molecule at the beginning can be broken off and added to an ADP molecule in order to produce our final ATP molecule of glycolysis and therefore complete the first of three processes in cellular respiration. So if you remember how we removed the previous phosphate from 1,3-BPGA, essentially what we did is we made a molecule that was so unstable that the phosphate within 1,3-BPGA wanted to break off and rejoin ADP in order to make ATP, and we are going to be doing exactly the same sort of thing. We want to make changes to the structure of 3-phosphoglyceric acid in order to make this phosphate as easy to remove as possible, only we're going to be doing it in a different way because we don't want to use ATP to add any additional phosphates to this. Now, the first of these changes is going to be relatively simple. All we want to do is take our phosphate, which right now is on carbon 3 of 3-phosphoglyceric acid, and all that we want to do is move it over here to carbon 2 in order to form 2-phosphoglyceric acid, or 2-PGA for short. And the name of the enzyme that we're going to use to do this is 3-phosphoglyceric acid mutase, and you can kind of see that the root of this is the same as the word as mutation, which refers to very, very subtle but permanent changes to DNA. And naturally, phosphoglycerate mutase is going to make the subtle change of breaking this phosphate off of carbon 3 in a hydrolysis reaction and then simply rejoining it onto this OH group via dehydration synthesis. And this was the reason why this OH group needs to be on carbon 2 with the double bond O on carbon 1 here. Otherwise, this movement of phosphate onto carbon 2 would not be possible. And as a consequence of this, not only is this phosphate here farther away from the positive pole on the H of the OH group here, but now this phosphate here is closer to this negative pole on the double bonded oxygen here, and we remember that this double bond is also a source of instability because if we have two bonds, that's 4 electrons that are relatively close to each other and therefore close to this phosphate, which generates even more repulsive force. Additionally, we also have the oxygen on the OH on carbon 3, so now instead of this phosphate having repulsion from only one direction here, because there's nothing on the left side here, now this phosphate is completely surrounded by repulsive forces on both sides, but we can actually do better and make it even more unstable. And that instability we can increase by taking this positively charged hydrogen, or this hydrogen on the OH group here on carbon 3, and we can actually get rid of this OH group entirely so that there is no positive charge that can stabilize the negative charge on phosphate. So here, and again, the reason why we want to get rid of this is that this positive pole can attract to the negative charge in the phosphate and stabilize that, but as we will see, our penultimate enzyme of glycolysis is going to get rid of this here. So the way that this enzyme works, which is an enzyme that we call enolase, or technically enolase, referring to a change in an alcohol, and the En gives us a hint that there's going to be an addition of a double bond in some way, because the suffix ENE refers to a carbon-to-carbon double bond. Well, there's no carbon-to-carbon double bond in this structure here, so what's the name, or what's with the name of this enzyme here? Well, here, the enzyme's job is rather unintuitively using its electrons to pull this hydrogen off. Now, this is very difficult to do because this hydrogen here is nonpolar, which means that this active site here needs a very specific structure in order to catalyze this reaction. Now, the two electrons that are in this bond here don't just magically disappear. Those are actually going to come down here because this carbon here is now missing a bond because we pulled this hydrogen off. So instead of reforming a bond with another atom, it can form a double bond like this, hence the name enolase. And the OL tells us that we're going to be doing something with this alcohol, this OH group, because now this carbon has 5 bonds, because remember that there are 2 hydrogens here already, and when we put this double bond here that used to be attached to this hydrogen here, now this carbon has 5 bonds, and the OL in enolase tells us that the alcohol, the OH group, is going to be broken off. And as a consequence of this, we have effectively removed a water molecule. So again, the enzyme initially pulled off this hydrogen here, but then the OH minus that breaks off that used to be this alcohol can take the hydrogen away from the enzyme, regenerating it, effectively showing us that we have removed water, and in its place, in the place where the OH group used to be, we have now made a carbon-to-carbon double bond and created the molecule 2-phosphoenolpyruvate. So we can simply abbreviate this as 2-PEP, phosphoenolpyruvate. Now, in terms of why we made this structure here, remember that this is all for the purpose of removing this hydrogen with a positive pole here, because that stabilizes the phosphate by giving its negative charge a positive pole to attract to, and now that positive pole, not only is that gone, but we have now added a second double bond, which means that we've got 4 electrons in a relatively dense position here, and all of that repulsive force means that the phosphate has become even more unstable, because now it's surrounded by this carbon-to-carbon double bond with no positive pole to stabilize it, but all of the negative charge on the other side from the polar oxygens with negative poles and from the 4 electrons in this double bond. All of this is going to serve to make this phosphate on carbon 2 even more unstable, and now at last we are ready to break off this phosphate. So the final phosphate here is once again taken by ADP. Let's actually spell that correctly. ADP, in order to make ATP once again. So yet again, we have another example of a kinase enzyme here. And the kinase enzyme here is what we call pyruvic acid or pyruvate kinase. Now, this name is a little bit of a misnomer because pyruvate or pyruvic acid is not the reactant molecule. It's actually the product molecule that we get. So more correctly, this should probably be called phosphoenolpyruvate kinase, but once again, we prefer the name pyruvate kinase because this is the final molecule that we're going to make. So once again, the job of pyruvate kinase is to remove this hydrogen here off of ADP in order to make this phosphate here reactive enough to want to attach to this phosphate here. And again, when this bond forms, it breaks this bond here off of the oxygen, which then goes to form a double bond like this. And then this double bond breaks off and actually takes the hydrogen that the enzyme originally took. And that's why here we have a CH3 molecule. If the mechanism doesn't appear to make sense, don't worry about it. The essential purpose here is that this phosphate here, because of the instabilities that we've created between this phosphate and the double bond and the oxygens on this side is strong enough to cause it to naturally break and join onto ADP in order to make our final molecule of ATP and end the process of glycolysis. In our final summary video, we're going to recap the entire process of glycolysis and take an inventory of the amount of ATP invested, the amount of ATP produced, the overall ATP produced, and an inventory of what other molecules we have that are products of the overall glycolysis process.